Really fun participating in this workshop, and I'm excited to see where it heads. And I will let's see, minimize this thing. I will tell you today about our work on hormones, and we're specifically about gibberellin today. But really, the way we're doing now is not just using our tools that we make, and in our case, hormone biosensors, to measure stuff, but also to try and understand what those measurements mean. And in the end, what we're arriving at is this idea that, just one measurement in isolation is only so informative, and really you have to understand how that measurement came to be as a result of a lot of different activities. And that's why we're talking about this idea of a symphony or an ensemble. Okay, so why is it not letting me go forward? So yeah, gibberellins is one hormone, but there's several hormones in the plant, of course. Most of you are probably familiar with them. They're very important in plant biology, but they're also very important in agriculture. This is just showing how, like, if you spray gibberellins on a grape plant, you'll get bigger grapes, right? So we all love this idea of hormones being plant growth regulators. And so what we want to know is a lot about how this all works in an endogenous situation and how we could better use it for agriculture. And so what we did when I was a postdoc was engineer a GA biosensor. And I won't tell you much details about how it works, but it's a FRET biosensor. So we have a GA sensing domain that interacts with gibberellin, and a fluorescence output domain that tells us how much the gibberellin is present in a soluble environment. And so we can express this protein, this one single holoprotein in plant cells, and get a readout of where the gibberellins are in vivo at the cellular level. And that might sound cool, and it is, and we really like to know that information, right? So we're looking at GA concentrations in a given cell. So already, like, a new technology, a new bit of information that we could add to something like a plant cell atlas. But where we are now in the lab is understanding that concentration of GA and the patterns and the dynamics of GA, we can measure them now, and they're very interesting. But really, that's the result of an ensemble of activities: biosynthesis and catabolic enzymatic activities, conjugation and deconjugation enzymatic activities. And because hormones are mobile small molecules, right? They move around cells, they move around the body plan of the plant. The transport steps are very important, right? And of course, all of these have regulatory steps on top of them, controlling their transcription, post-translational modifications, things like that. So really, we want to know about the hormone levels, but we also want to know about transcripts. We want to know about transporter proteins, regulatory proteins. And so we want to know about all this. And that's my vision for how a plant cell atlas could be quite powerful, is to start syncing these things up to understand what's really happening on a holistic level with these, complex systems that are emerging from component parts. Oh yeah, let me get this going here. Okay, so what we have is measurements, and this is on the left is reds are like high gibberellin levels in Arabidopsis root. And these are individual nuclei of individual cells. And then in the blues, purples are low gibberellin levels. So we saw this right away, this correlation with this GA sensor of GA correlating with cell growth, which I won't go into detail how we think the mechanisms of the gibberellins acting. What I'm going to focus on today is how do we get such a gradient? And of course, I mentioned that there's enzymes and transporters and their regulators. And if you look at this slide, not for the details, but just the overview, it's quite complex, right? So we have multiple subcellular compartments. We have multiple transport steps. There's a lot of stuff we know, some stuff we don't know. And so it's a huge complex system. And so where we are now is that we've been able to perturb the system, push the system in certain ways, to overexpress an enzyme, to take an enzyme away, and to see that we now think we have an idea of what is limiting GA biosynthesis and accumulation in this region. And we think it's actually largely driven by one specific enzyme and the expression of that enzyme, and a little bit by a precursor. And in the tip, right, in this region, something different, right? We think there's actually two biosynthetic enzymes, expression patterns, that are limiting GA in here, and that's why it's low here. And we found another really surprising result was just that in this middle region of the apical meristem, that a co-substrate might be limiting, which is kind of new information that the sensor once we started working with the sensor, we were able to reveal. So what we're doing now is asking can we put this together, all this information in a model to understand all the different components, and that's in collaboration with Leah Band at Nottingham. But this question of is the co-substrate limiting is really interesting to us. And unfortunately, we don't have the right tools to be able to perturb the co-substrate in different cell types, right, in different cell and locations. And we don't even have the tools to perturb the enzymes in, like, for example, these region only, right? So these kind of spatial questions and temporal questions, we're still limited by being able to push the system. And so that leads me into the next part of what I wanted to tell you about today in terms of new tools, is to be able to use light to manipulate biological activities in vivo at a cellular or even subcellular level. And you may have heard about optogenetics, and that's what I'm talking about, but optogenetics in plants is just getting started. And I'm going to tell you that we've been trying to co-opt this optogenetic system that was used in E. coli to move to work in plants. So it's called CCS-CCAR. It's a two-component system. It changes gene expression and in response to different light environments. And what we've been able to do is convert the system to be able to induce, for example, this NRC4R gene. So in red light, we can turn it on. In blue light, we can turn off the gene expression. And in what you're seeing here is defense-associated fluorescence, so immune response fluorescence. And so you can see there's a dynamic here. With red light, we see high fluorescence, and blue light, low fluorescence, whereas the positive controls are always high and the negative controls are always low. So we can actually activate immunity with light in tobacco plants transiently expressing our system. And this is pretty exciting because we can start to ask questions about what we can do in real plants and ask questions and maybe in the glasshouse or in subcellular level in the microscope. And what we've been able to find is that it doesn't need just all red or all blue. We can use modified white light with more red and modified white light with more blue to get gene expression on in the red enriched and off in the blue enriched. And these types of switches in light environment don't affect Arabidopsis development very much. So we might be able to then direct gene expression changes, whereas all the other genes in the genome won't be changing very much, right? So that's the idea anyway. So you can, and this is a general approach. It could be used for your gene of interest as well. And just because we have a very short time, so I'm wrapping it up already. But hopefully you have interesting questions, or any, if I haven't been clear, you can ask a question, I can clarify. But our biosensor toolbox is expanding. I told you about our gibberellin sensor. I didn't even really explain it, but we have many different sensors coming along the pipeline in the field, and many sensors from animal systems or fungal systems that can be moved into plants, right? Or even bacterial things. So there's a lot of different biosensors being developed, and they can permit all kinds of interesting high-resolution measurements of cellular activities in vivo, right? So what's actually happening in the cell. So even we can have now sensors for measuring phosphorylation events, things like that. And that kind of information, high-resolution information, is really powerful. And then the next step is to be able to do not just measure, but perturb, right? To understand how did everything get to be. And so that's the optogenetics that I'm talking about. And it's just getting started, but I think the future is very bright. And that we will have the ability in the future to be able to do high-resolution perturbations of cellular activities. In our case, we're trying to control gene expression, right? And that's general. So if we can control the expression of your gene of interest at high resolution, turning it on and off with light, and of course, light can be delivered with subcellular resolution. So there's quite a lot of possibilities if we can get this to work. So fingers crossed that we can the field. And I really do think our group and other groups are really working on this to make this a possibility in the near future. And I'd like to thank my group. Annalisa Riza is the GA biosensor lead, and Bo Larson is the lead on the optogenetics project. And